One of the things that we know today is that the viruses, bacteria, parasites, and other pathogens surround us and coexist with hosts like humans. And one of the things that we would like to understand is what differentiates specific pathogenic microorganisms from non-pathogenic microorganisms. So this leads us to this concept of a virome. Viruses are obviously some of the most clearly understood pathogens. They actually are not characterized as living microorganisms, but they only live in a host. They can only replicate within a host. And when we think about viruses, we can think about the virome, or the collection of all the viruses that are present in a specific environmental sample. Environment here can be the human gut, the human lungs, it could be the water that we drink, or it could be anything around us. And so virome or microbiome are these collections of microorganisms that we can sample and clearly identify. Now, within the virome or the microbiome, we can start characterizing the specific individual microorganisms. And one of the important things is that because of the next generation sequencing technology, we now have a variety of viruses, bacteria, parasites that are characterized at the genomic level. Now, the genomic revolution that originally started because we wanted to understand the human genome has in parallel led to an explosion of the number of viral, bacterial, and other types of microorganism genomes being sequenced and studied. And here you can see just a glimpse of how much information is available. And you can see here that the most studied viruses, for example, are the viruses that are commonly found to be pathogenic to humans. Now, obviously, we can kind of understand the logic behind this. For example, just if we look at the scale at which COVID-19 is being sequenced, we can see that the major outbreaks of infectious diseases like SARS-1, Ebola, Zika, MERS, and now COVID-19 have been these spikes in sequencing. Why do we want to sequence these genomes, and why particularly do we want to sequence pathogenic genomes? Well, one of the things that we want to try to understand when we're sequencing these pathogens is the specific recipe that these pathogens employ to be effective at what they do, which is to replicate, evade our immune response, and ultimately adapt to the different types of responses that typically work, but in their cases, they're not working, to overcome and continue and spread within the population. So these pathogens that we are going to study in this program are going to be studied using next generation sequencing to understand their genome. And then we're going to apply the same technology to understand the response. How does the human immune system respond to these different pathogens? How does it vary across different types of disease manifestations? And how can we characterize those interventions that we can design based on this interaction that we understand because of next generation sequencing data? So really the critical thing here is to understand this technology. How exactly does it work? But also, how can we analyze such data, and how can we translate the analysis to biological insights that are actionable, that are informative and actionable? So what is next generation sequencing? And now we're going to start with thinking about next generation sequencing in the context of pathogens. How can we use next generation sequencing to understand the genomic structure of viruses and bacteria? And how can we learn to identify important variation that we think leads to their manifestation of disease? How do they cause disease? Metagenomic and viral sequencing can be of several different types. Specifically, we can look at targeted regions that we already know. For example, ribosomal RNA is one way to look at only bacteria, because bacteria have ribosomal RNA that is different in structure from for example, human ribosomal RNA. And so that's how we can amplify a particular type of a sequence to identify only bacterial DNA or RNA. Another example for targeted sequencing is what we hear for SARS-CoV-2. When we only want to look at, for example, variants of concern that affect the spike protein. And so then we know how to amplify only the region that contains the sequence for the spike protein. So we can sequence particular regions by amplifying them, and that's called targeted sequencing. Another type of sequencing is total DNA or RNA. When we're not looking for any particular region, but we're saying, let us just extract all of the genomic material from a particular sample and sequence everything. And then finally, you can see here, amplicon environmental. In the same example, when we have a particular type, so one type targeted would be, let's target the spike gene. Another one, let's do a type of a sequence, and we'll understand the composition of a particular sample. Now, these are very different approaches, but they rely on the same technology. So to help you learn in detail, we recommend these three particular articles that you can read through on your own, and we'll share at the end of today's session the links to these articles that will help you understand how these types of sequencing approaches are helping us understand the presence and characteristics of different pathogens. Now, let's talk about sequencing specifically in the context of viral communities and bacterial communities, right? So microorganisms, again, pathogens. So a lot of times, one of the challenges with metagenomics, when we have an environmental sample that contains a lot of different present organisms, we would like to understand how all of these different organisms are characterizing the environmental sample. Now, in bacterial examples, there's always a challenge because with bacteria, we're not able to isolate and grow individual bacteria. Only 1% of bacterial isolates could be grown in the lab. So one of the questions would be, well, how do these bacterial communities exist in real life, in the real world? And to do that, we have the approach of metagenomics. With viruses, it's the same thing. Not only do we have viruses interacting with other viruses, these viruses also interact with other bacteria. In fact, the majority of viruses are bacterial viruses, viruses that only infect bacterial cells. So when we think of a particular virus that is causing a disease, we would like to understand the community or the environment where that virus came from. And in many cases, we would like to characterize that particular one pathogen, how that pathogen is different from the others that it lives together with or that interacts with others like them, and then understand how the sequence of that particular virus can be a piece of information that we can use. How can we use that piece of information? In viral outbreaks, we would like to take a clinical specimen, for example, a human blood sample, and we would like to find all of the viruses that are present in that clinical sample. For example, if we have a temperature, if we have vomiting, if we have cough, right? We want to understand what is the particular pathogen causing the manifestation of that disease. And to do that, what we can do is we can sequence everything that is in that sample that includes the host genome and maybe genomic material, as well as anything else that is present there. Sometimes we already know. So if we have a particular virus that we think is causing the infection, right? We can design a primer, specifically amplify those regions that we know should be present based on the virus strain that we think is present in this particular patient, and then try to understand, is that the same strain that we expect it to be, or is it somehow different from the reference that we have? Now, in many cases, we also have multiple causes of a condition, right? And so what's important at the first stage is to try to understand what all is present in a particular sample. And we can do that because we have different types of data where we can store the resources about potential organisms that we can find. For example, we have a database of microorganisms or a database of viral genomes, and we can now compare and contrast what we find in a sample with what we will find in a particular sample that we have, or what is known to be in a database. Now, to understand how these different techniques are going to be useful in the context of next generation sequencing, we have to understand the types of files